So it's been recording. The previous webinar series of last year is already available on the YouTube channel of the Midwest Grape and Wine Industry Institute. And so if you don't have the link or if you don't find the access to this webinar, I will send you the link on the chat box. So then if you have any question during all the presentation, I would like to have you to put your question in the chat box or the Q&A box. If you don't see any of them, you should have 3 dots at the bottom of your screen. And so click on the 3 dots and you should have access to at least the Q&A box. And so all the questions, as I said, will be answered at the end. So we will have 30 minutes at the end just to answer to all your questions. And also at the end of the webinar, you're going to receive an email with a survey, a link to a survey, and we will really appreciate to have your feedback just to know how we can improve our webinar series and how we can provide more information, more tools, more knowledge to you. Because I'm sure that's something you will probably use in the future in your vineyard or winery. So we would really want to have your feedback to improve ourselves. And so today, that's my pleasure to have Darren Michaels and Dr. John Moore from Scott Laboratories and Stellenbosch University in South Africa. And so they will present for 30 minutes about the theoretical and practical aspects of the enzyme selection. So that's going to be probably focusing more on vitis vinifera grapes, but that's something you can always start to use on cold hardy grapes. And you will be able to ask any question if you want about cold hardy grapes, how to use enzymes and what type of enzymes, the role of those enzymes on cold hardy grapes. And so on that, I would like to thank you both to be participating in this webinar today. And I leave Drew introducing our first speaker, Darren Michaels.
I'm very pleased and somewhat honored to introduce my old friend, Darren Michaels, who's now at Scott Labs. I knew Darren. He was a lab manager for Terravant, a wine company, a custom crush, custom label facility in Buellton, California. Darren was raised in California wine country. He got a Bachelor of Science degree in biochemistry from UC Santa Barbara, my old alma mater as well. He started in the work field by performing antibody fermentations at Scripps Immunology, before becoming a wine chemist at Trinchero Family Estates up in Northern California. He moved on to manage the Central Coast offices of Vinquery, and then became, as I said, lab director of enology and quality at Terravant Wine Company. Eventually, he fell in love with the Columbia River Gorge, and who can blame him? So Darren moved on to the Pacific Northwest, took a role with Lafor USA, where he spent 7 years as their technical enologist. Darren is an active figure in the wine community. He's taught winemaking courses for the Vesta program and also at Allen Hancock College in Santa Maria. Welcome, Darren.
Thank you, Drew. God, it's so good to see you. I knew Drew when he was at Lucas and Llewellyn years and years ago. But it's good to see you guys. I really like Midwest, and part of what I like about the Midwest is it's kind of, and Drew and I were talking about this, it's kind of an open world when it comes to, you know, wine, but also beverage, local consumption of wine. So it's really important, I think, honestly, for this talk, especially, is to kind of go over like some basic tools of what enzymes are, how they're made, and kind of maybe some techniques you could possibly use. The thing with, unfortunately, well, not unfortunately, just the way it is when we talk about enzymes, there's tons of enzymes produced, and so most of the ones you deal with in the wine industry are ones that were isolated for, that were developed for vitis vinifera. So a lot of the hybrid or non-viniferous varietals, we're not entirely sure oftentimes what the enzymes will do. I mean, overall, because Scott Lab's been selling enzymes for so long, and they have a lot of experience through, you know, organic experience or through some structural or trial-based stuff. So we do have some idea what works, but we're definitely not into, our company as a whole is really not into over-promising and under-delivering. And the thing with enzymes people should need to understand is that when you buy an enzyme, you expect an outcome. It's not something to where you buy an enzyme and kind of hope you'll get this thing. You know, with enzymes, you should expect and look for outcomes that are very specific. So we're going to try and work on that. Just really quick, when I was at UCSB, I was an undergraduate researcher, Howard Hughes Memorial Scholarship for polyphenol oxidase and ACC oxidase and per se Americana. So most of my experience has been deriving, isolating, and purifying those enzymes and doing kinetic experiments. So anyways, so what are enzymes? Well, protein with catalytic activity simply is what an enzyme is. So one substrate, one reaction. It induces or speeds up a specific reaction, and importantly, at the end of the reaction, it'll keep working until there's no more substrate or it's stopped. And that's kind of feeds into what we'll be talking about too. That's something you should kind of understand when it comes to enzymes. Wine enzymes are produced by fermentation or they're extracted from egg white. So because of regulatory limitations and also just European limitations, the vast majority of our enzymes that we use for the wine industry are based from Aspergillus niger, a mold. Most of them are pectinases, but there are some glucanases that we produce with trichoderma and Telleromyces. Trichoderma harzianum is okay. It has a different usage specification or regulatory concept for TDB. So some larger wineries don't like to use glucanases if they can because it doesn't apply, but we don't have to get into that. Egg white I consider to be our lysozyme enzymes that act against lactic acid bacteria. Those enzymes I don't really count as purified as much as extractions, enzymatically heavy extraction of egg white. In fact, my boss is the reason why we use lysozyme in the wine industry. She's the one that got it through GRAS, or helped get it through GRAS, and then eventually got adapted for the wine industry. I still really like lysozyme. It's one of my favorites for my own winery. We have a little winery in Tetrahedron called Tetrahedron in the Columbia River Gorge, and I do use a little bit of lactic acid bacteria or lysozyme because of our high pH wines. So wine enzymes are mainly pectinases with secondary side activities, glycosidases, and glucanases. So what are enzymes as part of their protein structure? The primary structure is usually about 100 amino acids. It's a chain of them, but sometimes if there's enough, they'll start to fold on each other because of the charges of the amino acids. It's kind of very simple in that they fold. That's called the secondary structure. And that brings in a third dimensional structural element. Now, if you fold it again, you'll get a tertiary structure. And that's where you get their specificity to the enzyme. So that's when the enzyme starts working. Now, if you have much larger enzymes, they fold again, become quaternary structures, and you'll have different structures like that. It's only for us because it also affects its efficacy. So bentonite, for instance, affects the tertiary structure of an enzyme. It doesn't remove it, but it does affect its activity. And something like high SO2 might affect the quaternary structure of an enzyme. So enzymes, industrial food enzymes. So there's tons of enzymes: amylases, galactolipases, xylanases, proteases, lactases, proteases, lipases, transglutamases. Different industries use lots of different enzymes. We have two main manufacturers that make specialized enzymes for us. Anytime you buy a Scottzyme enzyme or you buy a Rapidase enzyme, they're produced by two manufacturers. DSM, there's only so many enzyme manufacturers out there. There's 2 or 3. So when you buy an enzyme from a distributor, you might get a blend of enzymes that are from different, but we get ours directly from the manufacturers, and as a result, we get our specialized enzymes. So no one makes the enzymes we offer, but they're very close to other enzymes. The reason why I bring that up is Rapidase, which sounds like a fairly new name, is actually an old name. It goes back to 1920. And it's the first commercially available amylase enzyme. And obviously that was for alcohol production. As a lot of things, alcohol drives a lot of our industry and research drive, because all of us like to drink booze, or at least most of us like to drink booze. So when you're going to think about using enzyme, the first thing you want to do is see what kind of substrate you're trying to work on. So in general, for enzymes, you might, if you're using a starch, you might use an amylase. If you're trying to work on proteins, you use a protease. You kind of get the picture. If you add an ACE to it, that's the enzyme for the substrate, typically. So if you want something to work on cellulose, you'd use a cellulase. So if you want to drink if Drew wants to drink a lot of beer, he might be a beerase. Something like that. So the next thing you might want to do is select your type of action. So a lot of enzymes work on the ends of structures like exoenzymes. Not great for pectinases because these are large structures you want to break down. So you want a mix of enzymes that work in the middle of the structures and the ones that work in the ends. Then you also want to pick enzymes. This is not really important, but it's just kind of a general way for enzyme selection. You might select an enzyme that's active in acidic solutions, basic solutions, or neutral enzymes. For the wine industry or beverage industry in general, we're talking about acidic or enzymes that optimally operate in lower pHs. So by definition, we're looking at acid-friendly enzymes. But interestingly, there's also thermophilic or cryophilic enzymes. For the vast majority of time, most enzymes ideally work within a pretty much 45 to 75, 85 degree Fahrenheit range. But the enzymes like birapidase that isolated a protein stabilizing enzyme, that's a thermophilic enzyme called proteostab. And that optimally operates at 150 degrees Fahrenheit just right before it gets deactivated. And then they have a specialized enzyme that works really, really cold, below 40, and that's called clear extreme. That's a cryophilic enzyme. There's not very many enzymes at all that work at that temperature. Actually, KS enzyme is another good example. It's a proprietary blend of enzyme activities that works down to 30 degrees Fahrenheit. So it's very unusual that span of activity in most enological activities. But okay, so really make it simple. I want more stuff out of my fruit. I want it to work well. So you first pick your enzyme, the kind of enzyme you want it to work on stuff. You got pectin, you need a pectinase. Then really big thing is how will it work? Well, depending on your time, temperature, and dosage. If you're low on one, you can typically pick up on the other two. If you don't have a lot of time, like you're trying to depectinize for flotation or settling your must because you're worried about, or settling a juice because it's going to start fermenting, you might want to pick something that you could always increase the dose or increase the temperature. If you're at a low temperature, you're going to have to increase your dose or increase the time, and vice versa. For very large companies, they're worried about costing because the vast majority of them use enzymes. There's no way they can produce that much product without them. They'll look at lower dosages for costing. So when I work with a client like that, I'll be like, okay, well, you can't use this enzyme here, but you maybe could use that enzyme when you have a lot more time. So instead of using a specialized settling enzyme like Clear Extreme, I might recommend KS at a much lower dosage because that works throughout the age of the wine until they add bentonite. So there's different ways. It's just, those are the critical steps: time, dosage, and temperature. There are some things that stop enzymes or inhibit enzymes. Hard stop with bentonite, hard stop. So if you're and you'd be surprised how many times I talk to clients and we ask them about why an enzyme did or didn't work. I'll be like, well, did you add bentonite? They're like, yeah. And I'm like, well, then you're done with that. No more. So if you want to stop an enzyme, you'll add bentonite. Or if you've added bentonite to heat stabilize, don't add any enzyme until you've racked completely off of it. The other thing is high temperature, but most of the time wines don't get up to 180 degrees Fahrenheit unless you've done flash to taunt or you do some kind of pasteurization. And there are two other things that can do it. Very high SO2, I'm talking very high, 500 parts per million, much higher than you normally see. What that basically means is don't ever add your enzymes directly in a bucket with SO2. You can add it as much as you want, but none of the enzyme will work. So make sure you don't mix directly. But other than that, enological levels of SO2 work fine for most enzymes. The other thing can happen is if you add enological tannins. A lot of times if you add them directly together, because tannins bind with proteins, they might affect your tertiary structure of some of the enzymes. You're not going to get the best activity if you mix your enzymes with your tannin. As long as you kind of separate those additions, that are fine too. Two reversible things though with enzymes. One is low temperature. Okay, if you're really cold, You can always warm it up. Okay, it won't kill the enzyme, it'll just slow it down. And sugar content. There are specialized enzymes called glycosidase. DSM makes the largest spectrum because they're the ones that first isolated them. Rapidase makes Revelation Aroma, and that one will be inhibited by glucose, not fructose, but glucose at 5% or more sugar. But if you have an active fermentation and you're fermenting down below 5% glucose, it'll start to kick in and start working. Interestingly, the intracellular glycosidase, the stuff that's in the yeast, those are not affected by glucose, which kind of makes sense actually to me inherently, but it is kind of interesting. How do we make enzymes? Well, it's not much different from how we make a lot of other industrial kind of formats or fermentations, mainly driven by strain development. So if you have a good enzyme producer or manufacturer will select its strain, and that strain will be effective in making no acetylmalesterase, and it'll be effective in the enzyme activities you want. So you grow it up in a tank or in these big leveled fermenters, which are high surface area fermenters that grow a lot of cells and they grow a lot of enzyme. But anyways, you feed it the right broth or nutrients, and you can produce a lot of these activities. Glycosidase or Revelation Aroma is a good example of how they produce a strain with a lot of glycosidase activity, and then you produce a lot of glycosidase. So then they ferment it, then they isolate it, they separate it, they restore it, and then they concentrate it. Finally, they separate it. So you get a string of different kind of activities. But imagine it being a soup. It's very much like a wine in that way. So the cells have to be lysed. They don't extrude it. You have to be lysed. You get all the enzymes out of it. And after you get all the enzymes out of it, you might start to package. If you have to purify an enzyme, for instance, and it says that right there. If you find yourself going, oh, this enzyme is purified of acetylmalesterase, that's a problem, because what will happen is that along with the purification of removing acetylmalesterase, and we can get into that in a different conversation, it also removes a lot of side activity. So hopefully what you want basically is an enzyme that initially comes out as a liquid, and you have a choice. You can either get a liquid as an enzyme as a liquid. It's very easy to use, but it's a pain in the butt because you've got to store it in the fridge, or you can get you granulated. A lot of times enzymes that are purified have to be granulated because you can't get the liquid purified without removing some of the side activities. And generally, though, most people in the US like liquid. In Europe, they typically like the powder, but now Europe's getting more liquid. But anyways, long story short, the advantages are such that if you have a liquid, they're easy to use. They'll last about 2 to 3 years, depending on your storage, and you want to store them in the fridge. That's the problem when you're dealing with a huge amount of enzymes. You have to store them in the fridge. If you have granulated enzymes, though, whether they're purified acetylmalesterase, none of our enzymes have a lot of acetylmalesterase activity from their strain selection. So you don't have to worry about acetylmalesterase in any of the Scott enzymes, any of the ones we sell, because we're worried about that. But generally, even if you did get a purified one, or it's granulated because you don't want to buy as much liquid, you can get the same enzyme, but in a granulated form. And that granulated form is a little bit easier to store because you don't need to store it in the fridge. But either way, what I tell folks is, if you've got a little enzyme left over, and you've stored it in the fridge and it's liquid, it's probably fine. Just add a little more if it's past the expiration date. Okay, so why are we using enzymes? Well, really fast, the reason why we use enzymes is because you pay money. You're trying to make a boozy product that has flavor in it, right? You're not adding flavor. You can't do it with winemaking, but you can do it with beverages. So you have a product you're trying to make that make people want to drink, and they want to get a little bit buzzed. Then you have a raw material. Now, the raw material you're doing in the wine industry is very expensive. Grapes can be pretty expensive, $1,500 a ton, $2,500, $10,000 a ton. So the reason why you're using enzymes is because you're trying to take this stuff, the raw material, you're trying to get all you want out of it, and then so this can get better. Or you're trying to get your raw material, you're trying to get more out of it, so this stuff is not as expensive. So sometimes big companies use enzymes simply because their fruit is expensive enough is that they don't use the enzyme, they won't get enough of this product and they can't hit their price point. The rest of us use a little enzyme because we don't have the equipment to make the best wine we want. So sometimes you lose an enzyme because you don't have the way to make equipment. So with white wine making enzymes, we're typically adding enzymes to increase the aromas. And then you guys, can you watch my time? I've totally lost track of my time. Increase the aromas, increase the yields, increase the mouthfeel, or increase filterability in the same fruit. The reason why there's an arrow that starts on the left of your screen and goes right is because my point is that unless you add bentonite or heat the wine up to 170 degrees Fahrenheit, your enzyme's going to continue to work. It won't go away. It will run out of substrate in a white wine because you're far away from the fruit. So in white wine, you want more aroma, add some enzymes to the skin so you can get more of those aromas out. If you're trying to have a really good fermentation, there's not a lot of stinkiness to it. You might want to remove some of the solids you want to rack off, but you don't have a bunch of time or you don't have the refrigeration. You're going to use an enzyme to help settle, so it'll settle the solids. During fermentation, if you have any mold pressure, or if you don't think you have enough enzyme aroma or glycosidically bound aromas, you can add a glycosidase to help to release those aromas. Or especially by volume, if you're really worried about time frame for turnaround of your wine, if you're bringing the fruit in November and you need to sell that product near the end of December or the beginning of January the following year, you're typically adding some enzymes to help through the settle and clarification process. Why KS is such a very, very popular enzyme, because not only is it really effective in the wine industry, it's very, very effective in the beverage industry, because the beverage industry turnarounds are very, very fast. So in red winemaking, why do you add enzymes? That is either to increase the aromas, either speed up extraction with decreased tank turnaround time, increased mouthfeel, or increased filterability. So the bare minimum these enzymes should be doing is helping you quicken the time that you're going to ferment in the tank, and or also helping you filter or rack off. Those are the bare minimums. You can do a lot of other things, like a lot of clients like to get a lot of fruity aromas because most consumers don't want a whole bunch of tannin. So they'll get all they can out of the fruit. Then they'll press and crush because they need to get rid of that tank or get that tank turned over so it's empty. So you're getting a lot more aromas. It's a thing that you're trying to do, where I say enzymes are very much a process-based decision. You add an enzyme, you want something for that add. Enzymes should always pay for themselves. Okay, rarely will you add an enzyme and be like, good luck. You want an enzyme to either increase your filtration, lower your losses, increase the flavor of a wine or a beverage to the point where it tastes distinctly different from your competitors. So you're doing a lot of these things not just because it's kind of nice, but because you want it. And as a result, you should be able to see the downstream effects of it, So with white skin contact enzymes, they're very, very difficult enzymes to design, because you're adding an enzyme to increase the aroma or the flavor of a white wine, but you're not trying to increase any of the phenolics, any of the oxidized components. And if you do too much activity, you're going to end up with a mush, and you're going to end up with color, and you can end up with too much flavor. So it's very important to be careful on your skin contact aroma increasing. For Scott, you have Cuvée Blanc, Expression Aroma, and Sin-Free. Sin-Free is just a real great general use, very gentle extraction. You never have to worry about pulling too much of that stuff. But if you actually want a lot more aroma, you can use Cuvée Blanc, which actually has some glycosidase activity, meaning that it kicks in after the glucose drops below, but you're adding it because you want the final product to have more aroma. You're not just adding it because you think you might want to have more. Sometimes when the fruit doesn't have those aromas available in the pectin, as they are, for instance, Chardonnay. Would you have an aroma to Chardonnay? Probably not, because Chardonnay doesn't really have a whole lot of aroma to offer you. But if you have a Muscat and you're like, wow, I didn't really taste anything in this Muscat, that's a very unusual situation, because Muscat is literally strong enough to where they use the term Muscat light to describe wines. So you should be having a pretty strong flavored Muscat. You might look at enzymes for that kind of process. For large clients, you're typically using pressing enzymes specifically to increase the final amount of beverage you have from your raw material, in this case, grapes. So you're adding enzymes to quicken the press. You're adding enzymes to increase the amount of yield, and then moving towards settling, because a lot of residual activity, because you didn't stop it. You'll have a lot of enzyme activity that's left over that will help settle, but it's a much more processing decision. This white skin enzymes are more for a stylistic drive. Utilizing pressing enzymes is much more of a processing aid. Okay. Then there are specifically settling and flotation enzymes, totally different level of activity, totally different spectrum of activity. We're a little bit of wide range for his sources. It's very dependent on what people want. When I talk to them about settling enzymes, what they're kind of going for, but it's basically to get rid of your solids. Okay, so you can have a very clean fermentation. For cold soak enzymes, a fancy way of saying, do you want more aroma and flavors without too many polyphenolic extraction or too much tannin or too much whatever? You might use a very mild cold soak enzyme or something that's just specified for that. Cold soak enzymes and aroma enzymes for whites, cold soaking enzyme for reds and aroma enzymes for whites are the two hardest enzymes to design. That's why Color Pro is really, really popular for reds, because it's a very special enzyme because it doesn't over extract. So if you're in a huge, huge winery and you've got tons of processing going on and you have no idea how that fruit's going to be processed because you're not a genius, because you're a normal human being and you can't track everything, Color Pro is actually a pretty good enzyme for that, you know. But if somebody comes up to me and goes, I need to turn tanks around much more, then I would recommend either EXV or much more Color Pro. And the reason why I'd recommend those, this is for folks that want everything. They want to take their red fruit and they want everything out of that. Okay. The problem with doing everything out of your red fruit is you potentially could make it bitter. So I have clients that I go in and I say, look, what are your tanks like? So I have a client in Washington that turns their red fermentation tank 7 times. That's a lot. You know, usually you're fermenting once or twice, you're turning it twice, 7 times. The only way they can do that is they use specialized enzyme. That's a proprietary enzyme that we help them with. And that enzyme helps them turn those tanks around 5 days or 4 days fermentation without too much tannin, because they don't want too much tannin, but they want all the color and they want all the aroma. And that's a very hard enzyme to create for them. And it's a very client specific process. But because we work with manufacturers, it's a lot easier to do. So here's a flow chart to give you kind of an idea. There's Color Pro, Enzyme EX, after crushing, and then this kind of flows through some of the things you could do with enzymes. Sorry if I'm going a little fast here.
Darren, I'm just going to interrupt to let you know you've got about 5 minutes, 8 minutes left.
Great. Awesome. Hopefully I talked fast enough so no one can understand me. So release polysat. So in the final, you can also use glucanases, which is a specialized enzyme. Don't just add glucanase. You know, you want to talk to somebody who knows the regulatory status. Are you exporting your wine to another country? You might not be okay for glucanase. So if somebody says to you, oh, you could use a glucanase, you got to really be careful on that. But the flip side of that, especially if you're dealing with any kind of mold pressure, glucan is from botrytis or mold affected damage. And it's very, very hard to treat. In fact, a glucan structure is so complex that you can ruin a cross flow within like 3 months. So it's very important that if you're dealing with some glucanase or a glucan issue or potential, you might want to use some glucanase. The other thing about glucans that makes it very difficult, it takes a long time to break those structures down. So you have to use the glucanase very early on. The other thing you can do with glucanase is you can add it and it will actually help break down the cell walls. Sometimes when you run into polysaccharide finishing products, oh, this is a yeast-based product and it adds sweetness. They might have actually grown up some Lalon makes the vast majority of those kind of things. So they might grow up some yeast and then treat it with some enzymes and it'll release some of those compounds that make it very, very tasty in the finished product. There's other interesting product, which I'm not going to get into, which is a protein stabilizing enzyme. It's very interesting, but that's a specialized enzyme. But the one you can use right now, if you're really interested, is you can use a glycosidase. And Rapidase, Revelation Aroma, and Scottzyme BG are the two. Rapidase Revelation Aroma has the widest spectrum of glycosidases out there, so it's a good one to use. Scottzyme BG doesn't have as much, so it's kind of a different application. But the vast majority of time, you want to use this enzyme. Some large wineries do it a lot to increase aromas in white wines and even some red wines post-fermentation. If you're in the West Coast, you're worried about smoke, and some people use it for that. But bottom line is, if you do use it for that kind of application, you have to make sure you stop it, because all it's doing is hydrolyzing. And because low pH wine, pH is naturally low pH, so you'll be hydrolyzing those compounds over time anyways. If you use this enzyme, it's a very neat enzyme, you just gotta stop it. And finally, filterability. This is by far the vast majority of issues I run into typically this time of the year. Unfortunately, it might be late for clients. No, man, you should have added this way before. But when you're dealing with at Terravant, when I was directed winemaking there, we were trying to deal zero inventory winemaking, meaning like a client would fresh and easy be like, I want this wine. We're like, oh my goodness, I don't know if I can get you that wine. So we'd have to turn that wine around very fast. So a lot of the processing was very dependent on KS enzyme because we needed to get it processed and settled fast and go through the filter. I think that's, my goodness, did I hit it? I think I hit it. Was that too fast?
That's good.
That was good. There was a lot of information, and I'm sure our audience will have some questions. I've come up with a few questions myself that we'll get to in the Q&A. So thank you for that, Darren.
Sure.
Yeah, thank you very much, Darren.
Yeah,
I'm sure we.
Go ahead.
Oh.
No, sorry if I went a little fast. I just had a lot of information. But please, I recommend people rewatching this, or you can just call me or email me about anything you want.
Yep, I was going to say that too. We'll answer some questions at the end here. Those of you who are participating, who are viewing, I have to say this. I say it every time. If you've never called Scott Labs before, they're a very unique company. You dial their phone number, and an actual human being picks up the answers. And if you say, I have a yeast question, or I have an enzyme question, or I'm having a filtering problem, they will put you on to one of their experts, like Darren, or like Katie Cook, or any of the other many experts they have there at Scott Lab. They won't take your name and number. They will put you onto that person. And so you can have a personal, active interaction with any of the Scott Labs experts. And while I'm at it, I don't mean to be too pluggy. If none of you have a copy of the Scott Labs Fermentation Handbook, they are free. They come out every year. It's not just a catalog. They are filled with excellent research, suggestions of how to use their products. You can get the hard copy, or you can download it online. Again, I'm not trying to plug Scott Labs products so much, but they back it up with such excellent service and one-on-one service between you and their experts. So never hesitate to call Scott Labs, describe your problem, and speak to one of their technical sales associates.
People are standing by.
Aude, take it away.
Thank you very much, Darren, for the great presentation. Thank you, Drew, for the great promotion of Scott Labs. So I know that somebody is asking where they can find a hard copy of this catalog. And I think that's directly on the website of Scott Laboratories where you can request the hard copy.
Yeah, we're working on that. We should have the new one published very shortly. It was a long process.
Yeah.
But the enzyme section I put together. So make sure if anybody has any questions, let me know. But Drew is right. Essentially, yes. Call me. Just call me.
Yeah.
Just call me. Yeah.
Yep.
Thank you.
So now we're gonna move on a research approach with Dr. John Moore from Stellenbosch University in South Africa. So I'm sure it's pretty late now, but thank you so much to be with us tonight. And so Dr. John Moore is an academic on the staff of the South African Grape and Wine Research Institute in the Department of Viticulture and Enology at Stellenbosch University in South Africa. His research focus is aimed at developing cell wall profiling technology. Current research includes studying grape berry development, plant pathology, and the effects of water deficit stress from the perspective of the plant cell wall. Carbohydrate active enzymes involved in winemaking are also studied. He completed his studies at the University of Cape Town, where he obtained undergraduate degrees in biochemistry and microbiology and a doctorate in molecular and cell biology. His PhD thesis, entitled The Role of Polyphenols and the Cell Wall in Relation to the Desiccation Tolerance of the Resurrection Plant Myrothamnus flabellifolia, was performed under the supervision of the late Associate Professor Wolf Brandt. While under the co-supervision of Professor Jill Farrand, he developed a keen interest in the desiccation and drought tolerance mechanism of plants. During his PhD, he spent research periods at the University of Rouen in France, where he developed expertise in the biochemistry and microscopy of plant cell walls under the supervision of Professor Azzedine Driewisch. He worked as a postdoctoral fellow at the University of Edinburgh in Scotland in the laboratory of Professor Stephen Fry. And he's going to talk about the use of enzyme for deconstructing wine grape cell walls. So thank you so much, Dr. Moore, and the floor is yours.
Thank you very much, Aude. Can you hear me? Great. We're shifting gears a bit and moving not away from enzymes, but looking at enzymes in the context of grape cell walls. So the title of my talk, Deconstructing Wine Grape Cell Walls with Enzymes During Winemaking, followed glycoarray technology reveals new insights into Cabernet Sauvignon. So obviously it's not a hybrid, but we don't grow hybrids in South Africa. So just more or less what Darren was talking about. Well, what's the importance is that wine active compounds are locked up in grape tissues, and this is just a picture from Fontes et al., where they showed that phenolics, 20 to 30% of phenolics are locked up in the skins, and 80% of aroma is present in the skins, and then pulp accounts for only 10 to 20% of phenolics, and then more phenolics present in the seeds. So in order to access those compartments of the grape berry, we need to be able to break down those tissues. And important constituents of a grape cell is the plant cell wall. So plant cell wall models are quite complex, but they're basically composed of three major components, those being cellulose and hemicellulose, which is a neutral glucose-based polymer, as well as pectins, which Darren spoke about, which are the gummy polysaccharides that make the jammy constituency of preserves, and when you break down grape tissues, they form that sticky gel, and then glycoproteins, which are part of the cell wall as well. So if you look at the timeline of grape cell wall research, the first real work on grape cell walls was done in the 1980s by Luc Saunière, who's now at Nantes, but in France, but at the time was in Montpellier. And as the 90s progressed, more additional studies were carried out by Kylie Noonan from Adelaide University in Australia, looking at ripening of berry cell wall polysaccharides, and then enology studies by groups such as INRA Montpellier in France, Thierry Doco, and also Encarna Gomez Plaza in Spain, looking at phenolic extraction and cell wall composition. Now, many of these techniques that have looked at cell wall composition of grapes have used mainly monosaccharide information only. So they take a grape tissue, they hydrolyze it with acids, you release the sugars, and you look at the compositions of those individual sugars. But that approach doesn't tell you much information about the actual polysaccharides in their structurally intact state. So direct knowledge of polysaccharides is rarely investigated. This is inference based on monosaccharide data. So if you look at a complex series of polysaccharides, you can see in plant cell walls, we have cellulose, which is made up of one sugar, glucose. We have xyloglucan, which is made up of 3 to 4 different sugars, but mainly a glucose backbone. We have homogalacturonin, which is pectin, which is made up of a chain of galacturonic acid residues. And we have more complex pectins, such as RG1, which is made up of rhamnose alternating with galacturonic acid, as well as chains of arabinans and galactans. Now, if you hydrolyze all those polysaccharides together, you get a mixture of sugars, and you now have to piece it back together to figure out what is the polysaccharide that originated in the tissue. So one approach that can address this is to use antibody technology. And plant cell wall scientists such as Paul Knox in Leeds and Michael Hahn in the United States have developed monoclonal antibodies against a range of different plant cell wall polymers. And in the example here, you can actually use specific antibodies to pick up specific epitopes, which which infers the structure of the main polymer that's present in the sample. So what we did is I've collaborated with William Willits, a professor now in Newcastle University in the UK, but at the time was at the University of Copenhagen in Denmark. And we have all these antibodies available. So what we wanted to do was see, can we actually use this in an array format? And William's lab pioneered the use of applying antibodies to plant cell wall samples in using a microarray approach. They printed the arrays. You can print thousands of samples on arrays, and then you can probe them with different sets of antibodies. And here is an example of all the different antibodies that are available and the substrates epitopes which they recognize. So for example, there's 7 antibodies that recognize homogalacturonin and 2 antibodies against RG1, there's important pectin components, and then additional antibodies against xyloglucan and glucans and xylans and cellulose, as well as proteins such as extensins and AGPs. So with these antibodies as potential tools and the array technology being developed, you can actually probe samples, and you get an array like this, which we call the approach comprehensive microarray polymer profiling, or carbohydrate microarray polymer profiling. And what you get is you get a data set like this for your sample, where you have on the y-axis you have your sample number, Vine 1143, and then as you move along you get all the different antibodies that are binding to the samples. And in this case, you set the array to 100, to the highest signal being 100. It's a fluorescence detection method. And then you can normalize your array, and you can see that you can pick up in a sample, you can pick up whether the sample has got pectin polymers, hemicellulose or cellulose epitopes, and glycoproteins. And so this and you do a sequential extraction with CDTA, which is a chelating agent, which extracts your pectin, and then sodium hydroxide, which extracts your hemicellulose. So you get a rich source of data about which polymers are actually present in your samples and how those polymers vary due to various treatments. So as Darren mentioned, a lot of factors influence the choice of using enzymes and what the objectives of the choice of enzymes are. We know that things can be inconsistent in winemaking. So you have vintage variability, you have cultivar variability, you have differences in the maceration techniques you employ, and these would impact the objectives that you were aiming for when you were doing your winemaking. So were you trying to get increased color or increased phenolics? I'm particularly interested in the increased extraction of phenolics and color in grapes. So we are missing a key bit of information. If you look at the data the research up until this point. We went from grapes and we ended up going into wine and the color phenolics, and we'd measure things such as phenolics and color and tannins, but we didn't know what was actually happening really at the level of the cell wall. So we had some information, but it wasn't very thorough. So cell wall architectural data was really missing, and we needed that. It's tended to be quite a black box approach. And of course, what you put in determines what you get out, but why you get certain things out and not other things was not very clear. So we did a study that was supported by WineTech, our local wine industry funding body, as well as Lafort, based in France, in Bordeaux. And we looked at Cabernet Sauvignon grapes. And this is the work of Yu Gao, a PhD student at the time, who's now a researcher. And he basically looked at his main project outline composed of 3 objectives: to understand the role of the grape cell wall in the context of winemaking in Cabernet Sauvignon, and then to validate the cell wall profiling approach that we employed and the fractionation approach we employed in winemaking to get a baseline data set. Then look at the intra-vineyard variation and the impact of commercial enzymes, in this case from Lafort, and then the use of recombinant enzymes from Novozymes to understand berry cell wall structure. And then then I will conclude and then discuss future research avenues. So the first objective was a validation study, and that was to get baseline reference data sets of grape berry cell wall deconstruction and polysaccharide distribution during a standard red wine fermentation using this glyco-array approach. And so what we did is we fermented the wines, we added the enzymes following in this case, we didn't add enzymes, we added a settling enzyme. We wanted to see can we actually fractionate the structure and determine more detailed information on polysaccharide distribution within grape tissues in Cabernet Sauvignon pomace. So we milled the samples with liquid nitrogen. We did a sequential washing of the residue with ethanol and solvents such as methanol and chloroform and acetone. And we got a residue out, which we then probed with antibodies. So this is quite a complicated heat map that we got out, but the main idea is that we fractionate using different agents, hot buffer, CTA, sodium carbonate, and KOH, at different points in the fermentation. So at the start, the middle, and the end of the fermentation, and then using different extractant solutions, CDTA and sodium hydroxide. And we were able to show with additional monosaccharide data that there seems to be 2 main fractions that came out of the grape tissue of macerated grape tissue. There was a pectin-rich fraction, which contained a homogalacturonin or pectins with a high degree of methyl esterification. And we also got a hemicellulose-rich layer that came out. But this seemed to be coated with esterified pectins such as RG1 or raminogalacturonin 1. So we have 2 fractions, a more easily extracted pectin fraction and a pectin fraction that was more tightly associated with the hemicellulose. So we propose this based on the model, based on the figure from Noonan in 1998, which is just a microscopy image of a grape berry near the surface. You can see the skin cells. As you move further into the berry tissue, the cells become larger and thinner. So we think the de-esterified pectins, that's when you crush the grapes, the pectin tissue in the mesocarp gets released easily because this is highly de-esterified. As the grape ripens, those cells probably, we haven't proven this, but this is what we propose. Those cells become less methyl esterified, and you get two layers that remain behind in the pomace. And the first layer is a more easily extracted pectin component, which is partially esterified. But then as you get closer to the skin cells, you get a much more highly methyl esterified RG1 coated layer that's that's difficult to separate from the xyloglucan-rich cells and the cellulose. So in order to get to the skin tissue, your enzyme has to go through at least two complex layers of pectin. And the final layer of pectin is very tightly associated with this hemicellulose components. So we published this in a journal, Carbohydrate Polymers, where we discussed this model of two layers using the fractionation approach, two layers in the pomace tissue, and then we continued with the PhD study. The second focus of the study was looking at intra-vineyard variation. So we were interested in the fact that vineyards, our Cabernet Sauvignon vineyard, and the block that we were using to do our studies had variability. And we wanted to know how much variability is in the cell walls in those different grapes in the vineyard. And we have no cell wall data available on panel level variability within a grape, within a vineyard. So we investigated variation of grapeberry cell walls again for Cabernet Sauvignon on the harvest day, and we used a checkerboard design. And we wanted to add commercial enzymes and see, did they impact the variability of the cell walls during the individual fermentation process. So this is we have 6 vines in each panel, and we have 4 panels going across from north to south, and we look over 6 rows, and then we showed that every alternating panel was a control. So in the first block, U1 means untreated. Then the second block was treated with an enzyme, in this case funded through the 4, so we used their Lafay's Grand Cru enzyme. And then the next block was an untreated control. So each adjacent block became its own control for the different enzyme treatments. And we showed that the numbers are the Brix levels. So although this block area was supposed to be at 24 Brix at harvest, there was variability at the level of our subdivided panels. So it went from 19 Brix up until at the higher end 24 to 25 Brix, even though it was all supposed to be 24 Brix, in the ideal world. So what we did is we treated adjacent panels as controls and untreated sections. And we used 3 enzymes, Lafay's Grand Cru, Lafay's Fruit, and Lafay's XL clarification. So if we look at the monosaccharide data for the major sugars in the cell wall, we showed that looking at galacturonic acid, which is a very good indicator because that's the main sugar of pectin, if you had no enzymes, you had higher levels of galacturonic acid in your pomace material. But if you treated with control enzymes, commercial enzymes, in this case, in the figure, it's Grand Cru, Lafay's HE Grand Cru, you got a reduction in galacturonic acid, as well as an increase in mannose in the pomace material, indicating that some enzyme depectination had taken place. But interestingly, looking at the comp data, we could see that when treated with the enzyme, the backbone polymers of rhamnogalacturonin 1 and the side chains of galactans and arabidans, those signals decreased dramatically in the samples. More so, interestingly, than the antibodies that for homogalacturonin epitopes. So that was quite curious for us. So that indicated that something about the complex side chains of pectin in the RG1 form seems to be more important to have that component degraded than necessarily just homogalacturonin, Hg. And if we looked at it from so here we're using a PCA score plot just to see the variability of the samples of the CTA fraction, which is your pectin-rich fraction. So if you didn't depectinate, if you didn't add enzymes, the samples were quite widely spread and there was quite a lot of variation between the different panels. But where you added enzymes, that variation was reduced. And so the enzyme-treated samples on the horizontal axis indicates the reduction in pectin qualitative and antibody probe signals. Interestingly enough, in the horizontal y-axis, which is 10% of the variation in PC2, this is related to esterification levels of the Hg. And it shows that the enzymes do not affect esterification levels, at least in our data, which indicates, so usually if you have methyl esterification of pectin, one of the side effects could be that the methyl groups get removed and then you increase methanol in your samples, which is undesirable from a wine perspective. So clearly the enzymes are breaking down the pectin without breaking down the methyl groups of the pectin. At least that's what we think. Also looking at it from the sodium hydroxide fraction, which is your hemicellulose fraction, we could see that the enzyme-treated samples separated very clearly from the untreated controls. And this seems to be related to the presence of pectin RG1 and Hg homogalacturonin, again, complex side chains of pectin that are associated with hemicellulose. Whereas in the treated samples, these components were in much lower abundance. And then we looked at wine analysis after 1 year in the untreated panels versus the treated panels. And we could see that for color, phenolics, anthocyanins, and tannins, the levels were higher in the treated panels versus the untreated panels after 1 year of storage. So we published this in the American Chemical Society Journal of Agricultural and Food Chemistry, showing these results between looking at the intravineyard effect of adding commercial enzymes.
John, you still have about 5 minutes.
Okay, so the recombinant enzymes in winemaking was the third objective of the study. And in this case, we used various combinations of pure recombinant enzymes supplied by Novozymes. And we looked at 7 treatments: a no enzyme, endopolygalacturonase enzyme, an endopolygalacturonase and pectin methyl esterase enzyme, and a pectin lyase, and an arabinase and a galactonase, and Grand Cru as the control. And here I'm going to skip over this because this just shows where the enzymes act on the cell wall polysaccharides. Our main result showed that pectin lyase had a dramatic effect on the galacturonic acid levels and Grand Cru, but not the other enzymes. And in the CTTA fraction, again, the pectin lyase or the RG lyase rich fraction had an effect on breaking down the pectin, and the Grand Cru had an effect, but not the other combinations. So untreated and EPG had no effect. EPG and PME and arabinase and galactonase had deesterified. Pectin lyase and Cru showed depectination. Again, looking at the hemicellulose data, we get the same results. Pectin lyase and Cru gave clear results that pectin levels were being impacted by the addition of the enzyme. And we have, again, we showed that so pectin lyase and RG lyase and seem to have the strongest effect, even though we would think that endopolygalacturonase and pectin methyl esterase might have an impact. They did not have an impact. So it correlated very strongly with the Grand Cru control. And again, we had the same sort of results. In the pectin lyes treated and the Grand Cru treated, we got high levels of color, anthocyanins, phenolics, and tannins in our wines after 1 year of storage. And we published this work. So part one, we have from the study, we have two main fractions, a pectin-rich fraction and an esterified pectin-coated hemicellulose fraction. Enzymes not only facilitate depectination, but they also reduce the inherent variability of less ripe or more ripe, partially depolymerized grape cell walls. The pectin lyes and the RG lyes preparation proved to be the most effective depectination, and it may form a core ingredient of commercial enzyme preps. The endopolyglyturinase and the pectinyl methyl esterase did not work as well on depectination as we expected. So this activity may be inhibited by the wine environment or by the complex multi-layer component of the pectin material. So we developed a hypothetical model which might explain how the enzymes work in the context of winemaking. And we published this. And so we're looking at, as you move progressively closer to the skins, your pectin layers become more complex and tightly associated with the hemicellulose components of the skins, and they become progressively more difficult to break down. And so it's so in order to reach the good substances in the skins, you will have to have a preparation that's rich in pectin lyes and RG lyes, based on the work that we've shown in this presentation. But further work is necessary to confirm this. And so we've just presented this in a review recently, about 2 years ago, proposing that pectin lyes and RG lyes are your key enzymes in effectively breaking down the cell wall layers. Some other ideas are that cellulases may also work in concert with that, but that's still unproven. So thank you for your time, and I hope it's been useful.
Good. Thank you so much, Dr. Moore. That was very interesting from a research standpoint. I hope it wasn't too difficult, like too complicated for all of you that are with us. So that's the time where you can ask any question you have, either for Darren or for John. Feel free to use the chat box or the Q&A box. And I already saw 1 or 2 questions. And I think, Darren, you already responded, but I will ask them just to make sure everybody knows the questions. So I have made a skin contact Frontenac Gris wine the last 2 years that is very popular. Are there enzyme suggestions for skin contact wines that will help with aroma and not draw negative effects from the skins? Do you want to reply again, or?
Yeah. It was kind of a process answer, which is, if they're doing skin contact white, I'd be a little hesitant on adding enzymes in that step, because I'd be worrying more about over extraction. I suppose he could try possibly adding an enzyme post-pressing in the juice stage, maybe to help settle and maybe even potentially release some more aromas that might be stuck in some of the extra pectin he's releasing through that skin contact. But I definitely wouldn't recommend adding more enzymes to already a skin contact situation, because I'd worry about over extraction. Even a very mild skin contact like sin-free is kind of, I don't know. I'd be less inclined to add more enzymes to that situation, but he could potentially use enzyme in the must to see if that would help with some settling or some releasing some more of those aromas.
Mm-hmm.
My concern would be over extraction if he's already doing a couple or time in skin contact. Kind of depend on how much skin contact he's doing.
Yeah. Thank you.
Sure.
Another question. Just started working with Riesling last fall. Are there primary suggestions for this grape variety?
Well, my answer was, I do a lot of Riesling because I'm in Washington State, and it really depends on the style. Depending on the colder areas, you might have a more of a petroleum kind of classic German Riesling aspect in there, or is he trying to do more of a American consumer-friendly ester focus? So either one of those situations would require different steps, to be honest with you. But
And
you could
he
try
said more ester focus related to Riesling.
yeah. Then he could try and, but depending on like where it's like, I've made Riesling from Riesling fruit that's like, oh my goodness, it's just petroleum. So any steps I tried to take that were like, you know, ester focused steps, like maybe some anaerobic treatments, something that increases extraction with some skin content enzymes, or, you know, even trying, you know, ester friendly yeast strains with the right nutrition, all that stuff I did. And in the end, basically, I got the same Riesling I would have gotten anyway. So it really kind of depends. You know, if they're really large crops, sorry, there's traffic here. There's really large cropped and Riesling, the economics of Riesling production in Washington is crop heavy. So it's a little bit easier to make you know, maybe more fruit forward or those kind of maybe consumer friendly products. But yeah, depending where he's getting Riesling, he might just be dealing with that style. So, but tons of ways to do it.
Yeah. We've got another question on the chat box from Jean. What is the best enzyme for peach juice prior to fermentation?
You know, I think you know, here's the thing with enzymes and using enzymes in non-grape wines, because just like John had alluded to, there's a lot of research in wine, Vitis vinifera specifically, but it's limiting sometimes because as important as it is to do that information, it's sometimes for a lot of other non-Vitis approaches, you might have to take a different tweak. What I definitely recommend for peach, I might do a mix of PEC5L and HC, which is a hemicellulose heavy enzyme. It's more of a fruit processing enzyme that you might not see those activities in your typical wine enzymes. And the reason why I bring that up is because typical wine enzymes either were purified, or a lot of them are purified to remove cinnamylesterase, which is an active enzyme that could potentially produce off aromas depending on the yeast strain. I'm not going to get into that. But bottom line is, I use HC PEC5L. You might try just another clarification enzyme, but I wouldn't be surprised you won't be successful in that relatively more complex must. So that's what I would try. And then I wouldn't just use fruit enzymes, because fruit enzymes most of the time are not produced in such a way to have low cinnamylesterase production. So you run the risk of producing a heck of a lot of bad off aromas. So I usually just do HC and PEC5L for that. They can try that. You know, the thing that's nice about fruit wines is, you know, it didn't work. You know what I mean? Like, and so I'll be like, lean heavy in the beginning and be like, use like the top end, you know, for what kind of outcome you're looking. Typically, it's clarification, to be honest with you. But go ahead and use, and then if that doesn't work, you know, you don't have to try lower end, but you can always dial back. But with fruit enzyme or fruit-based wine production, especially like even seltzer production with fruit in it, you're really trying to get clarification. And yeah, you'll know it doesn't work because it won't be. That's what's nice about settling enzymes. The maceration enzymes and then the color-based enzymes are a little bit harder to verify the efficacy outside of very long-term trials. And even if you do those long-term trials, you might not see a cost-benefit analysis. You always have to do cost-benefit analysis. I bought this enzyme, it should pay for itself, right? It shouldn't just be a vague concept. So, but that's a good approach where you're like, no, it didn't work. And I'm like, yep, you're done.
Mm-hmm. Yeah, good, thank you.
Sure.
Drew, do you have any question? Or should I ask some questions?
I have some questions.
Okay.
From John probably doesn't have any experience at all with these these funny hybrid grapes that we use here in the Midwest, but I do want to ask a few questions directed specifically at hybrids. And I wanted to ask Darren his suggestions. Again, we do have high levels of pectin. We do have high solids in the white wine juices. So I wanted to get his suggestion on a good settling and a cold settling enzyme for white hybrids. And we also get some years we get very difficult to filter white hybrids due to a high level of glucans. And I wanted to see what his suggestion was for an increased filterability product for filtering some of these high glucan containing white wines.
Do you want me to go first, or do you want John to go first?
No, I was asking you, Darren. John,
Oh, me. Oh,
John doesn't have any hybrid experience.
Oh,
Really.
yeah, sorry. Yeah. Let's start with the settling. Yeah, so they're both basically settling enzymes. So you're right. Yeah. You guys with hybrids, okay? Yeah, you're going to run into issues sometimes with enzymes that were made for wine, because especially something like settling, because you're like, oh fudge. So you might even find that you use something that's okay in wine, or wine made from wine grapes, because and it's fine, but even at very high doses, you're not going to get the efficacy that you want. And the thing about the settling enzymes, even though you have a limited time, you could put it in a graduated cylinder and you could see if it's going to work. So the one practical enzyme that works over and over for settling has been, for us, one of two reasons. One is because of the wide range. So it's kind of like it's almost like a fruit processing based enzyme, but it doesn't have any cinnamolusterase, and that's kind of rare. It's fairly cryophilic, meaning that I can put it into a must that's actually pretty cold, and that would be Clear Extreme from Rapidase. But that's not surprising, because DSM is like literally the oldest enzyme company, one of the oldest enzymes company. So Clear Extreme is not something I typically offer to really large clients, simply because they're not at that state, but I do for flotation. But I would try Clear Extreme. And then I might lean on the heavy side, and then work your way down. And that enzyme is pretty at low temps, but you should be able to see it. Like you crush, you put it in a tank, and then you add the enzyme, and you should see it settle. So it's kind of a catch-22. You're trying to get it to get warm enough so the enzyme can work, but not so cold but not so warm that you want it to start to ferment, right? Because once it's fermenting, you're done. There's no settling. So, and I just dealt with that with a client where they're like, I've been letting it sit for 2 days and it's not settling. And I'm like, well, because it's fermenting, and by definition, it's not going to settle. So I would add it, and then probably around 40, 45 degrees is probably not a bad temp. Or you can risk it, crush the fruit, come in at 55, depending how cold. How cold are you talking when the fruit comes in? Is it cold or warm?
Again, Darren, many of these people are making less than a ton of grapes.
Okay.
They may or may not have a temperature controlled tanks. So I'm looking in very general, broad terms
if it's
here,
not
so
like
specific.
Yeah, if it's not 40 degrees outside, it's 50 to 55. Just use Clear Extreme. Use it at the high range. You can always dial back, and you should see settling. You should see it start to settle. That's probably the one I would go to. And I like Clear Extreme too. It's 100 grams, and it's powder, so it's never going to go bad. Stick it in the fridge next to the milk. You'll be fine.
Let me, if you will, let me describe what I suggest as a basic protocol. See if you agree.
Yeah.
Whether I'm using the Rapidase or just using a good old PEC 5.
Yeah.
I will add that enzyme during destemming and crushing, and let it work all the way through the entire press cycle. And once the press is completed and all the juice is in the tank, I might give that a half an hour or an hour for the enzyme to work. And then I would add some sulfur, if I've chosen to add some sulfur for cold settling.
Yeah.
I want to let that sulfur work for about 3 or 4 hours. And then I might add either Bentalact or just a small dose of bentonite if I'm worried about any harsh phenolics or any excess coloring.
Yeah, I like polycasyl because it doesn't have bentonite in that. It does have PVPP. But you could try doing this. You don't put the enzyme in there in the beginning because you're getting more aroma out of it, but it might be exacerbating high pectin content. You could try crush disemming, pressing. Don't put the enzymes until it's in the press pan. You add the enzyme in the press pan, and then you let it do its magic at hopefully at least 55, 60 degrees. Let that do its work, 4, 5, 6, 8 hours. Then you rack off. Then you can ferment on the polycasyl or Bentalact. That you could try that, because it could be that your enzyme might be actually exacerbating something that's limiting your settling. Does that make sense? Because if you're adding it in with the skin contact, it could be actually increasing enough. Now, the downside would be you might not be getting enough aroma. So it's definitely something to try to do. Crush the stem press, no enzyme. Add the enzyme as soon as it's pressed in the press pan or as you're collecting the juice. Let that do its magic, then rack off of it. Does that sound amenable? Yeah. Yeah. And then you had another question.
Yeah, what enzyme can help with difficult filtering issues?
Oh, okay, yeah. So glucans. Now I got it. Glucans are a pain. Now, for most of your students, you don't have to worry about exports. Let's just ignore that part. But glucanase, which is nice, Lauzyme MMX makes a really good glucanase, and it's in a powder. You don't need a bunch of it. But I would add it 1 gram per hectoliter or 2 grams per hectoliter fairly early on in the process. One reason is glucans are very complex structure. They're not like pectinase. You can add a pectinase to pectin and get fairly pectin native fairly fast, depending on your time, temperature, and dosage. But with glucan, no, it will take you a fair amount of time. So what I recommend, folks, if you have any mold pressure at all, is to add 1 or 2 grams per hectoliter. They do this a lot in southern France. 1 or 2 gram per hectoliter of Lauzyme MMX. You add it, during fermentation, even, especially add it during fermentation, so that it can start breaking those glucans down early. So by the time you do want to rack off that finished wine, you're already getting a better racking. Does that make sense? Now you can add both the glucanase and the pectinase, because the pectinase is not really produced in such a way. It's from Aspergillus niger. It's not going to have any. It'll have a little glucanase activity just from Aspergillus niger, just as a side effect, but it's not enough to really do a great deal amount of work. That's one way to do it. The other way you can do it is potentially add a wide spectrum enzyme like KS and then add it during the aging. But you need to build in enough time in your processing to have that glucanase. Weeks. So it's a real bummer to hear from clients, oh my goodness, I have a glucan issue. I'm like, when are you bottling? They're like, 2 weeks from now. I'm like, oh no, you're not gonna make it. But it's really, really important to know too that when you do those enzymes, even the regular enzymes, they're going to still working without the bentonite. So even if you can't do much with the glucans, you could try your best you can to remove the other pectin structures as well that will help the food processing, or at least break down a lot of them so they can settle. But settling enzymes are easier to deal with and easier to work with because you don't have to worry about over extraction. Whereas like John had seen, that there's a very gentle, very hard balance in extracting enough so you actually have a difference in your final product in red wine making, but not so much that it tastes like garbage. So it's definitely like, ooh boy. So yeah, John, I mean, like when we look at a wide spectrum amount of enzymes out there, there's some enzymes out there that literally don't do anything, but it's better than doing too much, except it's of course it's not worth the money because it's not doing anything.
Well,
Yeah.
one of the things that was really I was thinking about is skin thickness in your American hybrids. What is the issue with skin thickness?
We purposely breed grapes to have thicker skins and looser clusters, so they can take the insect pressures, the climate pressures, the temperature pressures. So we do have thick skins and thick pulp. It's a challenge.
And that's how,
Yeah.
so I had a question related to that, the cell wall structure, because, so John, you just explained that the cell wall material is different depending on where you are. So if you are in the skin or if you are in the pulp, the cells are different, the cell wall material are different. And so with our interspecific hybrids, we don't have the same thickness. And then we also don't know the cell wall material, what type of polysaccharides we have, what type of proteins, even though we know, we have an idea of proteins that we have. And in those berries, we are rich in proteins. And so that was two questions. So the first one is, what about adding enzyme? I'm thinking about extraction of phenolic compounds, because that's one of the thing we don't have in red wine, a lot of phenolic compounds. And so if we want to have more phenolic compounds in those grapes, should we try to identify a way just to break those skins before adding enzymes? And so, sure, but then adding enzyme, it just means you're adding another protein. So how is that gonna react with the phenolic compounds?
We haven't really seen, the enzymes are relatively small. You know what I mean? They're 100 amino acids or such. They're not super big. You know, I haven't seen a whole lot of, in a controlled manner, with or without enzymes, you'll typically see either not a really great effect to the enzymes, but I haven't ever seen, and then research had done before with Terravon, I hadn't seen a whole lot of, I've never seen it diminish color. You know what I mean? Which would be a definitely big giveaway. But it's like bentonite, right? Bentonite actually doesn't remove enzymes. They're not big enough. It will affect your tertiary structure, but it won't really remove them. So I don't know if but then again, I don't know about hybrids, but I don't know if you necessarily need to worry too much about the presence of the enzyme, but I would recommend the catch-22, I think, is the enzymes are not going to work on the waxy cuticle part, right? Like no enzyme work, right? You have to crack it. And then by definition, once you crack it, you know, then you're exposing the protein that's in the fruit to the phenolic. So it's kind of like the idea of maybe a sacrificial tannin. So I'd be interested in seeing official component initially go in at crush the stemming, and then see then potentially plus or minus enzyme, and then see if there's any ways you can stabilize those newly released protected compounds, either through micro-locks, and I'm not, you can do this in a trial, maybe not on a commercial scale, but or maybe doing a catech-enriched tannin to see where it's at. But yeah, it's a catch-22. Like, you definitely, it would help to crush it and then add the enzymes. But then, yeah, you're running the risk of, right? It's like you got all the protein there.
So I've been collaborating with Encarna Gomez Plaza from the University of Murcia in Spain, and one of the things that they've been testing is using different sequential additions of enzymes to see if they can actually break down the thicker cell walls, and not using just pectinases, but also using cellulases to see if that treatment might be more effective, with various degrees of success.
Was it on Cabernet Sauvignon as well, or was it on another?
No, it was on Monastrell, Mourvedre.
Okay.
So they're also making hybrids in Spain. So but it's not, it's vinifera hybrids, so it's not non-vinifera hybrids. Yeah.
And so the research you have been presenting, did you do the addition of enzyme right at crush, or did you do the addition of enzyme during, like when did you add the enzyme?
At crush. We
Okay.
followed the instructions from Lafort. So they wanted it at crush. So
Okay, yeah.
Yeah, we did back in Trinchero. This is dating myself here. When I was a wine chemist there. It was kind of a mixed bag. They use enzymes now, but it was kind of old school. It was 20 years ago. It was a lifetime when it comes to wine-based, enzymes, because they're basically fruit enzymes. And it was really interesting because we were doing, God, what was the index? We were doing, I can't remember of analysis. It was basically total phenolics. It was like real rough, phenolic analysis, right? But we found that the enzyme, we had a real mixed bag when it came to the enzymes and final products. Like we'd add the enzyme and then, and then this is before Color Pro, like specialized enzymes. So it's like, what we found was exactly what you said. We'd add enzymes, and some of the wine made from it would actually, we'd get more initial increase of some of the compounds, but then they would simply dissipate or go. And so the concept back then was, well, enzymes are just not effective. But what ended up figuring out was that protein chemistry. It was the binding. So even if we had an enzyme and it worked initially, and then it was vintage based. Like some years, depending on, the pathogen stress, these are pathogen related protein stress, or stress related proteins. It was like that year the enzymes did a ton of work. And then the next year we didn't do any work.
Yeah.
And I still really do think that still happens. We still have and I think something like a hybrid situation where we just don't have a whole lot of data. This is why I'm really excited about a lot of your students, because you guys get a chance to actually go down these rabbit holes, because it's out there. I think it's totally possible. Is that too much?
We've got a question from Hector. He was asking, there are many enzymes, commercial and brands in the market. When a winemaker wants to use one of them for white or red wine, which do you recommend? And specifically, I work with Grenache and I want to extract more color and aromas for my red wine. Which brand and type of enzyme do you recommend me?
Oh, well, I mean, I work at Scott. So it'd be Color Pro or Enzyme EX. Those are the two we'd have.
I've used Color Pro on Grenache many times, and Color Pro works great with Grenache. Absolutely.
Yeah, I used a lot of Color Pro before I worked at Scott. I've only worked at Scott since 2019, so yeah. Those are the two, but you could try other ones. It's exactly what John had alluded to when he said different enzymes. What I wouldn't recommend is an enzyme that is heavier on the cellulase and heavy. I would recommend ones that are specifically for where they say aromas or for, color, quote unquote. You don't want ones that are going to build mouthfeel, quote unquote. You don't want ones that are over extract. And so definitely, it's really more of a lack of than it is, more stuff you want. That didn't make any sense. That was super scientific, by the way.
Yep. Thank you. Do you have any other question? Because it's 4:31 and we were supposed to finish at 4:30. So if you want to stay for more questions, feel free to use always the chat box and the Q&A box. We can stay for a few minutes, I think. If you have to leave, just stay tuned for the future webinar we're going to have on July 6th. So every month we start to do those webinar series. And so we are going to talk about yeast selection next month. If you have any question we didn't answer or other question, feel free to reach out to Drew or myself. And remember to complete the survey. That will just take you about 2-3 minutes. So please just give us your feedback. I would like to thank all of you to participating in this webinar, and obviously Darren and John for participating. Thank you very much, John, to be with us late night during the night.
Yeah.
I hope you're not too tired, but do you have any other question? Anything? Anybody has any question? Drew, do you see any questions? Let's say that's all good, probably. What did you say, Drew?
I said, I don't see any more questions.
Okay.
I think we'll call that good.
Mm-hmm.
Again, I want to thank Dr. John Moore for working so late for us here in the Midwest, way in South Africa. We really appreciate it, Doctor. And Darren,
My pleasure.
and I'll just remind you all again. There's some great research and information in the Scott Labs handbooks. It's not just a catalog, and you can, if you have simple questions or questions come up you haven't thought of, you can always reach Aude or myself. Our emails are there on the screen. I know both Aude and I reply to all emails and very quickly. And thank you all for joining us this time.
Yeah. Thank you very
Thank
much,
you.
everyone. And it
All
seems
right.
that the survey link has expired, but I will fix that. Thank you.
Thank
Right.
you.
Take
you,
care,
guys.
Thank you,
everyone.
everyone.
Take care. Bye.
We stay in
Adiento.
touch.
Yeah. Call me
Bye.
anytime. See you
Yeah.
guys.
Bye. Thank you. Bye, Dr. Mo.